Good morning, students. Welcome back to my channel, Molecular Mysteries. So, in the last class, we have discussed about the male reproductive system, the location, the parts, and also the longitudinal section of testis. So, in this class, I told you I will talk about the spermatogenesis. What is spermatogenesis? Where does it happen? And what is the divisions involved? What are the cell divisions involved? And what are the phases of your spermatogenesis? For that, without any further delay, let's get into the class. Okay, hope you're all following regularly. But if you have any doubt, please let me know in the comment section. I'm also adding the link for your male reproductive system location and also the structure and parts, because you'll come across that again in this class, because you have to get to know about the seminiferous tubules. Where does that particular spermatogonia gets converted into the spermatids? So without any further delay, let's get into the class. So I told you this class is all about the spermatogenesis. What is spermatogenesis? Spermatogenesis is a formation of sperms, right? It is a formation of sperms, and sperms are the main important for your fertilization. So the testis, called the male gonads, produce sperms, and you have 200 to 300 million sperms produced per per day in human testis. Okay, that is how it is. Like, okay, you have 200 to 300 million sperms, and you have immature sperms which undergo maturation and differentiation in the epididymis of your male reproductive system. So let's discuss that in this class itself. So before that, we all already gone through this diagram in the last class itself. We all know these are the coiled tubulate structures called seminiferous tubules. These seminiferous tubules are packed in a compartment like structure called the septa of your testis. Okay, this is the location where your spermatogenesis happen. So for your understanding and for your easy understanding, I'm giving you the picture. Okay, hope you're all aware of the anatomy of your testis. In the last class I've covered. If you want, you can go and check it. Okay, this is how the seminiferous tubules look like, and here now I'm cross section, or I can say it's a cut location of your tubules, how it looks like under microscope. So how that really looks like, what are the parts of your cross section or longitudinal section of your seminiferous tubules? This is how it looks like. If you can see detail, this is the basal lamina, or I can say this is the wall of your seminiferous tubules, which have been taken for one longitudinal section, right? So you have many things like that, because they are all opening up into the lumen, the center, or the lumen, right? It starts with the spermatogonia at the back, or at the wall, or they can say the basal lamina, and it have various stages forming spermatids at the lumen of your seminiferous tubules. Okay, this is the microscopic diagram. It starts with the spermatogonia at the wall, ending up with the spermatid at the lumen. So it is for your understanding I have given you. So if you have any doubt, so you can also go through this. So this is the diagram that I wanted to talk about. Later I will also discuss what are the phases of your spermatogenesis, right? So before that, so this is your, just remember, this is your wall. Okay, this is your wall of your seminiferous tubules, and these are all the sperm types of cells that starts with spermatogonia, ending up with the spermatids at the base. This is called the lumen. Okay, so now let's see how it looks like. So for example, if we talk about what are the phases of your spermatogenesis. So I told you spermatogenesis is the formation of sperms. It has mainly two important phases. They are spermatocytogenesis and spermiogenesis. So don't get confused. Spermatocytogenesis. This is the one which starts with the basal primary germinal cells or primordial cells. They are the primary germ cells from where it starts. Right? These are located in the walls of your basal lamina of your seminiferous tubules. Right? So from where it starts multiplying, undergoing various types of divisions, right? So the spermatocytogenesis starts at the wall, and they are called the primary germ cell, or I can say primordial germ cell. This primordial germ cell at the basal lamina of your seminiferous tubules undergo various different stages, and finally produces four immature spermatids. Okay, these are all immature spermatids. They are not yet mature, and they are not yet motile. They are immotile and they are immature. Please remember. And the second phase of your spermatogenesis is called the spermiogenesis. So, which is involved only for the maturation and the differentiation of your spermatids. These spermatids that are produced at the primary step now enter into this spermiogenesis, finally forming a well defined matured sperm. But this matured sperm is not yet motile. It takes time for that to have a motility. So it undergoes that in the epididymis, which I will complete in the next coming slide. Okay. So these are the two important phases, spermatocytogenesis and spermiogenesis, that happen during the process called spermatogenesis. So for a glimpse I have given you. Now let's start about discussing about the first stage, spermatocytogenesis. What is spermatocytogenesis? I told you the primary germ cell or primordial germ cell now is a type A primordial germ cell. This primordial germ cell produces two different cells called primary spermatogonium type B and primary spermatocyte. Okay, please remember the primary spermatogonium undergoes division forming two important cells called primary spermatogonium type B, and you have primary spermatocyte. Okay, this division, why this type A B is produced, the purpose of producing the primary spermatogonium type B is to increase the number of your primary germ cells type A. That is why I am giving you a reverse tick here, so that this keeps on increasing the number of type A. Thus, this mitosis and meiosis keeps on continuing. Actually it is increasing the number of cells of primordial germ cell. That is why type B I have given you a arrow mark in the reverse direction. Okay, and now I told you it produces the primary spermatocyte. From primary spermatogonium, you are getting primary spermatocyte, which is a diploid in number. It undergoes actually the mitosis, right? Once the mitosis happens, you have primary spermatocyte. This primary spermatocyte first undergoes a reductional division called meiosis 1, where the chromosomal number is reduced to half. Thus, you have two haploid secondary spermatocyte, right? You got secondary spermatocyte from primary spermatocyte. Please remember, once you got your secondary spermatocyte, which are diploid in number, undergoes a meiosis 2. So you are all well aware that meiosis 2 is almost resembling the mitosis. So you have four different spermatids produced from two secondary spermatocyte, right? So hope I am clear with the concept. So you have four spermatids which are haploid in number, producing from the two secondary spermatocytes. This is undergoing a meiosis 2, which is resembling in mitosis. So this is the stage 1 of your spermatogenesis. And now let's enter into the second stage called spermiogenesis. Please don't get confused with that. Okay, second stage is the spermiogenesis. Now you got your immature sperm cells, or I can say immature spermatids, which are in the lumen of your seminiferous tubules. Now these immature sperm cells, or I can say immature spermatids, has got Golgi bodies in it. It has got centrioles in it. It has got mitochondria and the cytoplasm in it, but they are still immature, right? If it should get matured, this maturity is called, you call this spermiogenesis. This step of your spermatogenesis doesn't involve any cell division, please remember, right? This is the second step, and this second step doesn't have any cell division. It is only meant for your differentiation, maturation of your immature sperms into mature sperms. Now I told you, once the spermiogenesis happens, it produces matured sperms, but they are non-motile. I'll let you know how that happens also. Okay, please remember. What are the phases of your spermiogenesis? So in the last slide I have given you, there are mitosis, meiosis and all. But this phase of your spermatogenesis doesn't have any cell division. So it has only the Golgi phase, acrosomal phase, tail phase and the maturation phase. So what does this Golgi phase include? Actually, this phase has got many Golgi bodies in it, which has many enzymes. These enzymes are involved for the formation of your acrosome of your sperm. You are well aware of the structure. Okay, if you have any doubt, I will show you a short YouTube short where I describe the structure of your sperm. It has got acrosome at the top, right? So you have acrosome, and that acrosome is formed from the enzymes from your Golgi bodies of immature sperm. And you have your acrosomal phase, which condenses to form a cap. So once your acrosome is formed in the Golgi phase, that get condensed and form into a cap, at the pointed cap at the top of your sperm. And you have your tail phase. I told you already that centrioles in the immature sperm cells will get elongated, finally reducing itself into the tail. That how tail is formed in a matured sperm. Now I told you the maturation phase. Once these are all ready for formation, this particular immature sperm cells has got much amount of cytoplasm. This cytoplasm get finally loosed or loss of this particular cytoplasm, leading to the matured sperm at the final part. Though the spermiogenesis ends with the matured sperm formation, which is non-motile. These mature sperm, non-motile sperms will be formed in the lumen. It will be there in the lumen. They finally enter into the epididymis, where the spermiation takes place. The spermiation is the gaining the motility for your sperms. So for that to happen, it has to stay in epididymis for a few days. There the motility starts. Right? The gains motility in epididymis, and finally, once it has got the maturity, once it has got the motility, this sperm is eligible for the fertilization. It is having that capability to swim towards your female reproductive system. Right? This is the capability of your fertilization. So this is how, see, please don't get confused with the terms that I'm using. Spermatogenesis is the production of sperms. This spermatogenesis has got two important phases called spermatocytogenesis and spermiogenesis. Spermatocytogenesis is the production of immature sperms. Spermiogenesis is the production of non-motile matured sperms. And maturation and formation of motile sperms into from the mature sperms is what you call spermiation. See, you have three different: spermiation, spermiogenesis, spermatogenesis, and spermatocytogenesis. So if you have any doubt, please let me know in the comment section. Hope I am clear with the concept. If you have any doubt, please let me know. See, this is how it starts. It's spermatogonium. I told you primary spermatogonium in the walls of your seminiferous tubules. I told you, yeah, serotolys cells. These are the supporting cells which help for the nutrition of your growing sperm cells. Please remember, the serotolys cells are the I can say they are the supporting cells. They help the support, and also they provide the nutrition required for the conversion of your primary spermatocyte into secondary spermatocytes, finally spermatids and spermatozoa. Right? This is the location of your spermatogenesis happening in the seminiferous tubules. So, if you have any doubt, please let me know in the comment section, because in the coming class I will also talk about. If you want, I can talk more about the sperms and also the fertilization, embryo formation, if you want to get to know about that. Okay, so I told you only the sperm which has got high amount of motility, which is only one sperm fuses with, though there are millions of sperms produced per day in the testes, only one sperm is fused with the egg, right, which leads to the embryo formation. So if you have any doubt regarding the female reproductive system, I will also attach them in the description box. Please let me get to know more about that. And if you have any doubt, please let me know in the comment section. Hope I am clear with the concept. See you in the next class with an interesting topic. Thank you and have a nice day.